To test the interaction between two proteins, the two genes of interest need to be cloned into our BiFC vectors using gateway cloning technique. These constructs are transformed into Agribacterium JoVE3101. The JoVE3101 bacterial cultures containing two constructs will be mixed and co-infiltrated into the N-Basmiana leaves. If the two candidate proteins interact with each other, the YFP signal can be observed using a confocal microscope.
The main purpose of BiFC technique is to test interaction between two proteins in vivo. We have created a series of gateway-compatible BiFC and Easter2 hybrid vectors, which provide researchers with easy-to-use tools to perform both BiFC and Easter2 hybrid assays. In this video, we are going to demonstrate the ease of using our BiFC system to test the protein-protein interactions in an Basmiana transient expression system.
First, you need to prepare the JoVE3101 bacterial culture. You need to pick a single colony from your plate and inoculate in 5 ml of YEB medium with appropriate antibiotics. Then, you need to shake these cultures overnight at 28 degrees. The speed is 200 rounds per minute. The bacterial cultures should be ready the next morning. Make sure the bacteria didn't overgrow.